Hey, welcome back. I'm going to give a brief lecture on membranes and transport. So if you have your book, this is chapter 8 in your book, and as always, here's my email address if you need to get a hold of me. But essentially for this lecture, I'm going to be talking about the membranes and how things are transported across those membranes at a very high level. So as we know, cell membrane separates the living cell from non-living surroundings. So it's a thin barrier about 8 nanometers thick, and it controls things going in and out of the cell, but it has selective permeability. So it allows some things to go across the membrane more easily than others. Some of these things that are hydrophobic may pass easier because of the hydrophobic nature of the membrane. And again, the membrane is made of phospholipids, proteins, and other macromolecules, as we've talked about. The membrane is a collage of all these proteins, molecules, and it's embedded in a fluid matrix of the lipid bilayer. So you have glycolipids, transmembrane proteins, peripheral proteins, cholesterol, phospholipids. All these things make up the membrane, and some of these are involved in transport, as we'll find out here as we move forward. Just as a brief outline, I'm going to review membrane proteins and their involvement in transport, membrane carbohydrates, a movement across the cell membrane. We'll talk about the difference between active and passive transport and end with some relevant examples of transport systems. And again, the last slide here is your review, it's your study guide for this lecture. Membrane proteins are, as the name describes, proteins that are integrated into the membrane of a living cell. The protein itself determines the membrane's specific functions. So cell membranes and organelle membranes can each have unique collections of proteins depending on what they need to do. That membrane proteins can either be peripheral proteins or integral proteins. So the peripheral proteins are those that are just loosely bound to the surface, such as this one here, and it contain cell surface identity markers used for research purposes or for clinical purposes. Whereas integral membrane proteins integrate within the membrane, they penetrate the lipid bilayer, usually across this whole lipid bilayer, and they are often referred to as transmembrane proteins. They can also be transport proteins such as channels and permeases and pumps, all of which we'll talk about in this lecture. As we've talked about before, there are different types of amino acids that give protein structure. The important ones for this lecture are really the nonpolar and hydrophobic, as well as the polar and hydrophilic. And the reason for that is the polar and the nonpolar hydrophobic are within the membrane, whereas the polar amino acids are pointing inside and outside and inside the cell. So the hydrophobic components anchor the protein into the membrane, and if it's a peripheral membrane protein, and it may only be integrated in very top of the membrane, for example. Whereas the polar amino acids are extended into the extracellular fluid and into the cytosol and react with different things. As a way of a couple of examples, water channel bacteria is shown by this pore here. This helps shuttle water in and outside of bacterial cells. There's proton pumps, which we'll talk about. This one, for example, is in photosynthetic bacteria that can pump protons across a membrane. These types of proton pumps will become very important when we talk about oxidative phosphorylation and the electron transport chain, which I'll cover in a separate lecture. But these can shuttle these molecules through functional or through conformational changes by essentially changing the shape of the enzyme or the protein. And we'll talk about how that works here in a few slides. We also don't want to forget about membrane carbohydrates. These also play a key role, and these play a key role in cell recognition. So cells can distinguish themselves from each other using the various antigens on the cell surface, which are often carbohydrates that are bound to the membrane. These are also important for organ and tissue development, It's the basis for rejection of foreign cells by the immune system. The ability to donate blood to different people is because of these membrane carbohydrates, as we've talked about in the past. Okay, let's talk about the movement across. Let's talk about the types of transport processes. You can have simple diffusion. In the case of water, this is called osmosis, where no energy required. You're simply moving a molecule from an area of high concentration to low concentration. Something called facilitated diffusion is very similar, although you need a protein to help you along, in this case, to move from high to low concentration. But again, no energy is required. Both of these are called both of these are examples of passive transport. This is in contrast to active transport, where you need energy because you are moving from an area of low concentration to high concentration. So you're moving against the normal gradient. So again, passive transport can be either simple diffusion or facilitated diffusion. So simple diffusion is just diffusion of nonpolar hydrophobic molecules such as lipids. You're going from high to low concentration. Facilitated transport uses a protein channel or some other type of protein molecule. And again, going from high to low concentration, no energy is needed. Whereas active transport is diffusion against the concentration gradient. It requires ATP. It uses things like protein pumps, or instead of ATP, it can use an electrochemical gradient, which we'll talk about. So we have the passive transport versus active transport. First, I'm going to talk about normal diffusion. So diffusion is basically the second law of thermodynamics, where the universe tends towards disorder or entropy. Molecules move down a concentration gradient, as we discussed. So if you have a concentration of molecules of a dye, for example, and across the membrane, it's going to move across this gradient from high to low concentration until it reaches equilibrium. And at that point, the equilibrium, there's no net movement of molecules. Things are still moving here, but the concentration remains the same on both sides. So obviously the cell membrane is the boundary between the inside and the outside. It can't just be an impenetrable boundary, but we can't have just anything going in and out. So we're obviously taking in food from carbohydrates, sugars, things like that, and we're moving waste products out. So the point is here, cells need materials in and waste products out, and we have to move across the membrane to do that. So some molecules can get through by simple diffusion, some of the fats and other lipids. Some salts and sugars may require facilitated diffusion or some sort of active transport because they're more hydrophilic in nature and can't just diffuse right across that membrane. Very large molecules, starches and proteins, cannot move across these membranes at all. So those are very compartmentalized. Some examples of diffusion in biology: there's gas exchange in the alveoli. So oxygen from air, and oxygen is received from air to blood, and carbon dioxide from blood to air. There's also gas exchange in photosynthesis. We get carbon dioxide from the air to the leaf, and then oxygen from the leaf to the air. There's gas exchange for respiration, oxygen from blood to tissue cells, and then carbon dioxide in the opposite direction. Transmitter substance such as acetylcholine from the presynaptic to postsynaptic membrane at the synapse. I talked about that briefly in one of my early lectures when we talked about botulinum toxin. This is an acetylcholine transfer as an example of diffusion. The rate that determines diffusion are based on mainly these four things. First, the steepness of the concentration gradient. So the bigger difference between you have one side of the membrane to the other in terms of concentration, the quicker you're going to have diffusion across that membrane. Higher temperature also speeds things up, so you get quicker movement across that membrane. The greater surface area you have, diffusion can spread out across that membrane. So this is the greater surface area you have, the faster the diffusion. And then obviously the type of molecule that's going across or the ion that's diffusing across the membrane. Large molecules are going to need more energy. They're going to be slower. By energy, I mean more kinetic energy to get them across the membrane. And nonpolar molecules diffuse more easily than polar things because of the hydrophobic nature of the membrane. So that's diffusion. Let's talk a little bit about facilitated diffusion. So the facilitated diffusion, again, is the movement of specific molecules down a concentration gradient, so from high to low, but it passes through the membrane via specific carrier protein or channel. So it either uses a carrier type that grabs the molecule, flips it across the membrane, or the molecule moves through a channel. For example, some peptide antibiotics can act as ionophores to increase the permeability of the membrane to specific ions. So that, for example, there are two types: mobile ion carriers such as vancomycin. This can bind potassium, allowing it to diffuse across the membrane, and channel formers as well, such as gramicidin. But let's go back, take it back up a level for a minute. So again, this is diffusion through protein channels, and these channels move specific molecules. However, no energy is needed, as we've talked about. So carrier type proteins work by cycling between conformations in which the solute binding site is accessible on one side, and then there may be some sort of intermediate. So it grabs the molecule, there's some sort of intermediate, and then dumps it out the other side. So with carrier proteins, there's never an open channel. You can see this is closed at all times. It's open on one end, grabs it, and then opens up on the other end. This is in contrast to the pore channels that we talked about that are open on both sides. So that's passive transport. Let's talk a little bit about active transport, where the cell actually needs energy to move molecules against the concentration gradient. There are two main types: the primary active transport systems, which use ATP directly to drive the transport. So they're taking ATP, using hydrolysis of that to move things across the membrane. And then there's secondary active systems that use electrochemical gradients such as potassium or hydrogen ions, or a membrane potential produced by primary active transport processes. And we'll talk about each of these main types. First, we'll talk about primary active transport systems. Some examples are ATP-dependent ion pumps. These are grouped into various classes based on transport mechanism. One of the main ones is the P-class ion pumps. These are a gene family that have a very similar sequence homology. A very specific example of that is sodium-potassium ATPase in the plasma membranes. It uses an anti-port mechanism, and we'll talk through each of these types here. But it catalyzes the ATP-dependent transport of sodium out of the cell in exchange for potassium entering the cell. Some other examples of P-class pumps, specifically these in the stomach, we use some hydrogen potassium ATPases. These are involved in acid secretion. Calcium ATPases are found in the endoplasmic reticulum and plasma membranes. Some other examples besides P-class pumps, there are ABC, ATP binding cassette type transporters. There are a ton of these in nature which catalyze transmembrane movements of various organic compounds, including amphipathic lipids and drugs. We're not going to discuss these here because there's a lot of them, but they all use this primary active transport mechanism. Those were examples of active transport systems that use ATP directly to drive transport. Another type of active transport, as I mentioned, is secondary active transport. These use electrochemical gradients or membrane potentials produced by primary active transport processes. They can be either uniport, where they're moving one molecule across, symport, where one molecule is going in and the other molecule is going out, or excuse me, symport, they're both going in, whereas antiport, one's going in and one's going out. These are different. These are active transport mechanisms because one of these molecules is at least going against the concentration gradient. So uniport again is a single molecule, carries transport of a single molecule across the membrane. An example of that is the GLUT1 glucose carrier. This differs from the symport, which again, two molecules are going in the same direction, and then antiport, A is going in and B is going out. The symport or co-transport carries two dissimilar solutes or substrates and transports them across the membrane. But the transport has to be coupled. So the gradient of one substrate, usually the ion, may drive uphill, so that's why we have this classified as an active transport system. The example of that would be glucose, sodium symport and plasma membranes. So you're carrying both glucose and sodium across the membrane. Other examples include lactose permease and the hydrogen symport carrier. With lactose permease, which is a hydrogen symport carrier carrying hydrogen and lactose. So these are pretty simple concepts. For the exam, I imagine this just to have a couple questions from this lecture. There's only really a few concepts here. The main one being this summary slide here, where we have the two types of passive transport being simple diffusion, which goes from high to low concentration, simple and facilitated diffusion. The difference with facilitated diffusion is you're using a protein to help you. The active transport, you go against that concentration gradient and you're either using ATP or some sort of electrochemical gradient to drive that. Just going to end with some examples of transport systems. Your salivary gland, the salivary gland duct actually plays a role, has transport systems. It plays a role in saliva secretion through these transport processes. So there are sodium-independent chloride bicarbonate anion exchangers that are involved in these processes. They generate ion fluxes in the salivary secretion. One notable clinical syndrome is Sjögren's syndrome, which is a disorder with these anion exchangers. It's typically an impaired expression of these exchangers, but it's essentially what it is an autoimmune disorder in which the immune cells attach and destroy the exocrine glands that produce tears and saliva. So if you're not producing saliva, tears, you're obviously symptoms are going to be dry mouth, dry eyes, some dry lips, mouth sores, etc. And I'll leave you guys here with some nice pictures to look at of what Sjögren's syndrome looks like. Hopefully you don't come across any of these in your practices. The last slide here is just the study guide for Chapter 8. You guys can look through that and let me know if you have any questions. Thank you.